laboratory for functional optical imaging. Hang on, I didn't turn on my microphone, apologies. Okay. And so I was really fortunate to be here in a time where optical sensors, here, this is a voltage-sensitive dyes in a Langendorf perfused rat, where you can see a sort of a vortex pattern here, small organisms, in vivo models, green fluorescent protein, all these different things allow us to sort of get a handle on what's going on in actually living tissue. And so I dedicated the last nearly 20 years of my life to developing imaging systems that could kind of harness that information, right? That could measure as many things at once as quickly as possible in 3D if I could, and in lots of different in vivo systems. And I'm not going to cover all of these here, but that's my overview. And then I wanted, especially early in my career, to have kind of a scientific question to sort of actually couch some of these approaches. And so I got really interested in the relationship between neural activity and blood flow. When I was training, it was in the early days of functional magnetic resonance imaging, fMRI, and we saw this weird signal in the brain, and we kind of didn't really know what it was coming from, and it combined, you know, physics and optics and oxygenation and metabolism. And so I got really into studying this relationship from a mechanistic standpoint, as well as to kind of inform better how to interpret the images that we get in functional magnetic resonance imaging. And so I wasn't really going to talk too much about this, but as I mentioned this morning, we got into lots of conversations about vascular smooth muscle. So I threw a couple of extra slides in here. So this is not microscopy, but this is another technique that I actually dearly love that we call wide field optical mapping. And so what you can actually do is you can take a mouse and open its brain and actually just shine LEDs on its brain, and you get obviously a picture of the surface of its brain. And the vessels that you can see on there are going to be different colors because they have different levels of oxygenation. You can also take that mouse and do a genetic modification. So we have a green fluorescent protein, which is something that comes from jellyfish. You're all aware of this. You can put that into neurons, and now your neurons will glow green. Now you can modify that green fluorescent protein so it becomes calcium sensitive. Okay, so what that means is that neuron will now flash happily to tell you that it's firing because there's an increase in intracellular calcium. Okay, so this technology has been around for a while, and we'll talk a lot more about this at the microscopic scale. But what we realized with this technology was that we could also image this on this mesoscopic scale. So you can express this green fluorescent protein, calcium-sensitive green fluorescent protein, in the excitatory neurons of the brain. You can actually thin the skull, you can expose the whole surface of the brain here, and then you can actually see all of these great regions of the brain. And by imaging using a light that will excite the fluorescence, as well as light that will be absorbed by the hemodynamics, you can actually create images that will show you not just neural responses, but hemoglobin, oxyhemoglobin, deoxyhemoglobin, and total hemoglobin. And so what happens in the brain is when you flick this animal's whisker, this region lights up to tell you there's neural activity. This is the whisker barrel. And then you'll also see this vascular response that corresponds to this increase in blood flow that is triggered. And so this signal here in the deoxyhemoglobin is what you would detect if you were doing functional magnetic resonance imaging. I bring this up simply because this is some of the fundamental research work that I've been doing where, you know, first things first, it's really odd that the brain does this, right? You've got neurons firing, and then you've actually got these blood vessels dilating. And people thought originally that these hemodynamic signals we were seeing represented a consumption of oxygen because the neurons were using energy. But actually what you get is this very reactive vasodilation, right? Which comes from vascular smooth muscle. So I figured I had to talk about it a little bit here. And so one of the contributions that we made was actually staring really hard at the way that these vessels actually dilate and realizing that that signal had to be passing along the vessel somehow. And so I learned all about vascular endothelium, and we designed some experiments to see whether or not the signal actually could be dependent upon a propagation pathway that went along the vascular endothelium. So we did experiments like this where we actually shine a laser light to sort of cross the vessel here. That laser light actually stops this endothelial propagation signal going along it. We can truncate that. We can actually flood that whole surface with this light. We can cancel out that vessel signal, and we can completely change the hemodynamic response. Now, you know, this may or may not be especially interesting, but we recently got this funded by the Leduc Foundation, because one of the things that's really interesting here is nobody had really thought about endothelial cells, which are really a big part of your cardiovascular system, having all that much to do with your actual brain health, right? And yet we know that endothelial health is related to things like cardiovascular disease, obesity, high salt diet, smoking, all these horrible things that we all do. No, I don't, but so this is really kind of a potential mechanistic link between the sorts of things that we see in the body and the sorts of things that we see in the brain, and offers potential, therapeutic targets, for example. And we already actually know that some cardiovascular drugs have very good benefits for the brain. And so this is an area in my lab, actually, that we're quite actively exploring. We did similar work looking at neonatal development. Some of the earliest studies with fMRI found that there was actually negative bold signals in the developing brain. And so we also did a sequence of studies applying this technology to looking at postnatal development in the mouse brain and finding that this neurovascular system is actually something that's developing postnatally in the mouse. And so actually initially, when the neurons are firing in the mouse's brain, nothing happens, and you actually get hypoxia. And then over time, gradually that system kind of sets up and has this preemptive vascular response. And we even think that perhaps this hypoxia that you see at the beginning might actually be part of what's driving the vasculature to sort of know where it needs to be built. And so we've, like I said, as a physicist, I sort of plowed into some of these sort of biological and physiological questions because the tools that I had allowed me to see these things, and then I had to learn the biology to figure out what we were seeing. And really quick, this one is a nice example as well. So we realized that, somebody was talking about this a bit earlier, that glioma cells actually can migrate along blood vessels. And so we put a tumor into one of these mice. And one of the first things we recognized that we were seeing these little interictal events. And so we could really see these kind of sudden spontaneous flashes of activity. We could see that those were also evoking this hemodynamic response. That actually in the tumor, the way that hemodynamic response was being orchestrated was altered by the presence of the tumor. And so we just got this very exciting data where here we actually captured a spontaneous grand mal seizure in the mouse. And so you can actually really watch the neural activity here around the seizure. It starts with a bunch of these interictal events, builds into this massive tumor event. And this method lets us not just look at what's going on from the neural perspective, but actually even in the normal non-tumor bearing region, you can see that the vessels sort of dilate as much as they can, but then they really can't do anything else. And you start to see an increase in deoxyhemoglobin, which actually suggests there's a sort of hypoxia even in the normal tissue being generated by the seizure. And in the tumor, it's actually got such an altered relationship to the vasculature, it causes vasoconstriction and massive hypoxia within the tumor. Okay, so anyway, I felt like I just wanted to give you guys a little bit of an overview of some of the other stuff that we do. And so here with this platform, we can actually really look at the mouse's behavior. We can now map the neural activity of the mouse in real time. And we're using this actually to explore a lot of different relationships between disease, drugs, how the animal actually behaves, and how all of that is represented in its brain. Okay, so that's it. That was my little extra slides that I shouldn't have added this morning. I'm sorry, I couldn't help myself. Okay, so from here, so I've introduced to you now this idea that we can do optical measurements of neural activity. What I'm going to talk to you a little bit more now is about how we can take that to the level of imaging this kind of activity on the scale of single cells. Okay, so why do we need this fast 3D microscopy? Okay, so I've already talked to you a little bit about this calcium-sensitive fluorophore. So, first things first, right? Fast cellular processes. So why do we need it? Because we want to be able to see fast cellular processes within intact 3D tissues and organisms, right? Tissues that are not intact don't tend to be terribly alive, right? And almost everything that's alive is 3D. So if we really want to actually understand what's going on in an organism, we really need to be able to capture these things. And this is a good example. This is a zebrafish larva that's having basically epileptic seizures. And they're spontaneous, right? They're happening and they're coursing through the brain. And if I was just imaging one little piece of it, I would never really be able to see the way that that was actually orchestrating itself spontaneously in real time. Living things also move around, and so being able to actually image them while they're moving around instead of calling that motion artifact means that you can study things. So here are a few more examples. They also signal, so if we can actually capture them moving around and signaling at the same time, then we can really start to ask interesting questions. And then finally, something I hadn't even really appreciated is that there's a lot of really good structural imaging we can do now. We have clearing techniques, we have expansion techniques, and yet it's really boring to image these tissues. It takes a really, really long time. There is actually a stock image online of microscope boredom, which is someone who develops microscopes makes me really sad because it should be fun to use your microscope. But this is a piece of expanded brain tissue from Ed Boyden's lab. It's 4 millimeters long, and we imaged the whole thing in just 8 seconds. And so we realized actually that we had something if we really focused on going as fast as we possibly could, we could even solve questions of imaging dead things because it's boring waiting a really long time to image things. And also people, so we'll get to that. Okay, so, all right. So we want to make a fast 3D microscope. We're going to do 5 minutes of physics, so act accordingly. So if I want to image this three-dimensional volume, this is actually a living mouse brain. There's a dye in the vessels, and then the yellow color is astrocytes. A normal way to do this is using what's called point scanning two-photon microscopy, which you basically make a little focus spot, and you scan it around to every single location one after the other, right? And that's how you form this beautiful image. But this image took us about 5 minutes to collect that three-dimensional stack, right? But what we really want to do is we want to understand how those neurons and vessels or everything are interacting with each other throughout that three-dimensional volume on a much faster timescale. So we built one of these two-photon microscopes, and we pushed it really hard. We got it to the point where we could do about 100 frames per second, which was almost at the limit of what we could detect. And then we did this calculation. We said, if we need to measure every single one of these spots one after the other, in a 400 by 400 by 100 pixel volume, that's 16 million points, right? And my laser that I can use for this imaging pulses at 80 million pulses per second. And so even if I do 1 pulse per pixel, I will never be able to image this volume any faster than 5 volumes per second. Plus, that's only 1/7 millionth of the mouse brain. I would be scanning my scanner 10 times faster than any available scanner that you can buy. And even if I wasn't using a pulse laser, my pixel would only see the light for 12.5 nanoseconds, which is very close to the fluorescent lifetime of green fluorescent protein. Okay, so it didn't stop me convincing a student to try to build this thing, but after we did this calculation, we were like, oh no, this is not good, right? Like, if you have to visit every single one after the other, we simply are not going to be able to get to the point where we can image very fast in 3 dimensions. So this technology, light sheet microscopy, sort of popped up on my radar. There was a seminal paper in 2004 that showed that it could be done to image zebrafish. It was early used for imaging cleared tissues here. And then some of the really exciting work here, this was looking at Drosophila embryogenesis, right? And so this tells you some really important things, that light sheet is capable of doing really nice 3-dimensional imaging. And the fact that this embryo is not shriveling up and dying also tells you that somehow light sheet is quite gentle, right? It's not actually causing a lot of phototoxicity that's killing the sample. But what you can't really see very well here is that you just watched 7 hours worth of data, right? Because this imaging system's imaging 1 volume every maybe 30 seconds, right? And for a developing embryo, that's fine, right? You want to be able to run it overnight and then deal with this enormous amount of data. But so I wanted to figure out whether this technology that seems sort of counterintuitive to me was something that we might be able to leverage to push to really high-speed 3-dimensional imaging. Okay, so why is light sheet different from what I was just saying? So when you do light sheet, you create literally a sheet of light through your sample, and then you take a camera and you basically focus onto that sheet, all right? And that gives you what we call optical sectioning. So when you're doing the point scanning, you're just focusing your little spot on one particular location. This would be like me kind of illuminating a whole row of you from the side and then focusing on you from the front. And that means that I can just isolate just you as a row, and I can image you all at once. So I can get this equivalent of sort of optical sectioning in 3D by creating this light sheet. And I can make pretty good resolution there. That resolution is sort of the product of the illumination and then how basically tightly I'm focusing on you from the front. Okay, and this has been shown to cause way less photo damage. This is a measurement from Tim Hawley's lab that shows he can do 20,000 frames with his light sheet method, whereas if you do confocal microscopy, after about 200 frames, there's no fluorescence left. Okay, so we spent a lot of time trying to figure out, why? Why is it so much better, right? And part of it is because if I come and illuminate you guys from the side, you guys in the front and in the back aren't getting light on you, right? But if I'm trying to illuminate you all from the front and kind of focus on you, you're all getting blasted all the time. Okay, so I can just selectively illuminate this plane, and some people call this technology selective plane illumination microscopy. So I can selectively illuminate it, so I'm only bleaching the plane that I'm imaging. That helps. But there's a few other things as well, if I'm using a camera and I'm imaging all 400 by 400 pixels of that plane all at the same time, I have 160,000 times longer to collect signal. Okay, so this is a little bit like instead of meeting every one of your graduate students one after the other, you have a meeting and you all meet at the same time, right? So they all get to spend longer with you. Maybe it's not as productive, but in Lightsheet it's helpful, right? And so that means that you can actually put way less power because you can have a lot longer to collect the signal that you need, okay? And so it also means you don't have to scan like crazy. You can collect one whole plane at once and you just have to scan once to get that whole three-dimensional volume. The other really neat thing, and I just like this, so I'm just gonna say it, is that when you put the light in this forward direction, that light only has one chance to have a meaningful interaction. Whereas if I put it in a sheet, if it doesn't interact with the first person, then it has another chance to interact, and another, and another, and another. And so what that means is that you actually can get way more fluorescent light excited per photon that you put in. So I did a lot of modeling of this just to figure out if this was all really true, but this basically is the physical reason why Lightsheet is able to image really well with good signal to noise, but also without causing all of this damage that your standard in vivo microscopy is going to be causing. So it sounds fantastic, right? But then how do I now make it really fast, and why did nobody else make it fast? So one of the big limitations is that the way that you do it like this, if you have the light coming in from one side and then you image from the other side, then in order to actually create a 3D image, you have to actually somehow move everything, right? So you physically move your sample up and down, or you have to physically move your objective up and down. And anytime you're moving things, then you're going to have a big problem. Okay, so, and you also have to be able to hold it to move it, and that, living things don't really like that very much. You also have all these lenses around the outside, and so a lot of the systems that you currently can use Lightsheet for have this very complicated sort of ways of holding the samples and hovering them in between these different lenses, and it puts a lot of limitations on the kinds of lenses that you can use. Okay, so some people said, okay, maybe I can rotate it like this, and now I can image something that's sort of on a slide here, and in some cases, that can work, but it limits the depth that you can get in here, and it doesn't solve that problem of being able to do very high-speed imaging. So the technology that we came up with was a very odd idea, and I really couldn't even explain to you like how and why we came up with this crazy thought. It's the beauty of having, I don't even know. Anyway, so we thought, okay, what if we illuminate the tissue with an oblique sheet of light, and then we detect the light back through the same objective lens? So that immediately, so it's still a light sheet, right? But now immediately I don't have to worry about all those lenses around. I can now image something that's just completely flat or intact, like a mouse brain or my tongue or something like that, right? But I have to do something weird. I have to be able to somehow create this weird angled focal plane, which doesn't make a lot of sense. But, if I could then move that sheet from side to side somehow without physically moving the objective of the sample, then I would be able to capture this three-dimensional volume in virtually any sample that I wanted, it's sort of inverted or upright. And I would still be getting the advantages of Lightsheet, and I would be able to do very fast imaging. And so we came up with this very simple design. And again, I promised only 5 minutes on the physics, so we're almost done. This is very similar to sort of a confocal microscope. The light comes in, it focuses down here, then it comes back and it goes over here. But what we found was that we only needed this one little mirror here. We could use this mirror, and by moving the mirror, we move the light sheet back and forth. But because the light that's coming back from the sample bounces off this mirror and that corrects all of the rays, then I can actually always have a focused image of that sheet on my camera. And wherever I move that mirror to, I'm both moving my focal plane of my camera and my illumination at the same time. And so nothing limits how fast I can go except how fast I can capture my images on my camera. And so I can just move my mirror at 20 hertz and collect data at 20 volumes per second. And so this geometry allows us to, as I said, image all kinds of cool things. The data comes out in this sort of very weird way. It's like this oblique plane that's sort of like going backwards and forwards, but all you have to do is stick it back together. There's no reconstruction, there's no deconvolution. It's a genuine optically sectioned image that I'm just capturing very, 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 very quickly. And because I'm only measuring as many rows on my camera as I want depths into my sample, I can read my camera out really fast, on the order of thousands of frames per second. And even though that sounds like very fast, my integration time is still much, much, much longer than I would get for point scanning. Okay, and then the system itself is surprisingly simple, like really simple. Like we put this one in a suitcase and we took it to Dresden in Germany and used it for a course. It fits exactly in the trunk of my Subaru. We've taken it all over the place. It's a little laser, just a 1 galvo mirror here. This is completely isolated, the objective lens. There's no electronics or anything. There's like one wire that goes into the system. We do dual color imaging always. And so it's actually also relatively inexpensive, certainly compared to any other technology that tries to do super high-speed point scanning. And so we actually entered into licensing agreements with Leica and ASI, and at some point you'll be able to buy one, hopefully. In the meantime, I actually didn't put this slide in, but we spent really the last 10 years really working with people, helping people to build these systems, supporting them to use them, and I'm going to pretty much now give you a big rundown of kind of like what we've been doing with this and what we've learned that we can learn with this kind of imaging. Here's a little example of us actually using the system. So this is scanning a zebrafish larva that's just sitting there. These are literally those planes of its brain. This is it sitting in a little dish under the objective lens. It's just a standard 20x 1.0 NA water immersion lens. And that's it. We were imaging this fish for an hour, and that's Venkater. And he's like, "What are you doing?" And I'm like, just ignore me. 1.5 terabytes an hour on the slow system, about 6 terabytes an hour on the big system. What can we image? So I'll just let this one play for a minute. It's like, these are all like my children. I can't not show you all of them. So some of these I'm going to tell you a bit more about, and some of them, like this little tardigrade, we're not going to talk about, but literally, it's been so fun and nice to have people from all over the world sort of come and go like, I just could never have imaged my sample without this, and so taking especially our system to courses and things, we've learned a lot about what it can do, and so what I want to give you now is some vignettes on some of the different samples that we've studied and what we've been able to learn. So number one, maggots. He tells me not to call them maggots, but they're maggots, right? So fruit fly larvae, my collaborator Wes Gruber is really interested in proprioception in the fruit flies. And there's been massive investment into fruit fly science over the last sort of decade or so. We've got all these genetic lines, we can label all kinds of different things. And when he first came to me, he said, I can label these different proprioceptive neurons down the body of the larva, right? So these are the neurons that basically sense movement. And so his student, they put the calcium indicator in them. So they're like, okay, so now what we need to do is actually see how these proprioceptive neurons actually encode movement. But his student Rebecca was putting them on ice and squashing them between two pieces of glass, and then these larvae were like squirming around, and of course they couldn't move, but she had to slow them down because she couldn't image them quickly, right? So we built this little rig, just a little channel for the larva to just crawl, and we were like, go larva, crawl, and we just imaged it, So this was some of it wasn't the first imaging, I'll admit. This was like many iterations of not having such nice images, but so these are the dorsal proprioceptive neurons, and this is literally three-dimensional volume collected at 10 volumes per second. So this is looking down on it from the top, and this is looking at it from the side. And just simple things like the way that kind of those dendrites crush as the animal is moving was something that, hadn't been observed. That's not correct. This is supposed to be the ventral side. Okay, so the ventral side also looks really cool in that case, and we can stop this movie at any point in time and you actually see the way that these things kind of flip up into the body. This is just structural imaging, okay? But what we can then do is express that calcium indicator, so we can do red as a static indicator and the green as a calcium indicator. So now what you can see is actually that you can see these actually brightening of those dendrites. You can see these little axons lighting up. And so now you can actually capture this whole dynamic system as it's behaving. And so then what you need to do, so it starts to get complicated, you have to do tracking. So now you can track these neurons in three-dimensional space as a function of time, and so that actually gives you the behavior. It tells you at any moment in time, this is the distance between the different segments, and because you've now tracked them, you can also now extract their calcium signal, right? You have to be careful, because if you extract the calcium signal, it's also going to be changing a little bit because the tissue's compressing, so that's why we have the red label as well, so we can ratio the calcium signal to the red and make sure that we're not getting any kind of artifacts. And then from there now we can actually watch, as a function of how all of these different segments compress, how the calcium signaling is encoding to the animal, like where its body is. And so we first did one neuron type, and Wes was like, you know, well, it's a shame we didn't do the others. And I'm like, do you have the maggots? And he's like, yes. And I'm like, well, let's do it. So it turned out, so what he'd been working on actually for years was trying to understand the patterning, trying to understand why these different types of proprioceptive neurons reached out and kind of put themselves in all these different positions, right? And we knew from the ablation studies that if you kind of knocked one of them out, you know, the larva, like, went a bit slower, but there wasn't really any idea of kind of what the point of all of them was. And so by imaging all of them and kind of lining them up based on, you know, when the actual segments were compressed, we got this result, which we also sort of were like, okay, but then we lined them all up. And what we thought was really interesting was that actually each one of these different proprioceptors was actually set up to monitor its own little special piece of the way that that movement was occurring. And so when the larva's crawling forward, they're all like, okay, something's happening here, okay, now this one, now that one. And it sort of explains, it's like having the sensors on your car that kind of make sure everything's okay. So if you knock one of them out, you know, something's like not quite okay. But this tells you not just sort of the fact that they're encoding all of that, but what was super interesting is these all have these special different shapes so that they're anchored to things that tell them about stretch or tell them about compression to capture all these different nuances of the animal's movement. And so my little foray into neuroscience here was actually extracting those signals and generating a little model. And this is really sort of not very tidy looking, but so this is the original data. So these are the two neurons at the top of the head, and this larva is doing what it wants. It's just doing this. And so just based on the signals from the neurons, I was able to generate this like fake version of the larva and actually really decode the way that those signals were actually talking to the brain. So just from this, it's really interesting relationship of, it was like the sum of them versus the difference between them was really telling the larva where its head was in terms of being up and down and from side to side, which is actually physics, frankly. Okay, so that was maggots. So fish, I've already shown you this epilepsy video. This is just to show you that every single individual dot here, we can actually read out exactly as a calcium firing pattern of every single one of these neurons. We can do analysis of, for example, oh dear. Sorry, sometimes the videos go funny. We can do analysis here of exactly where this seizure started, right? And we've traced all the way back and study how that, for example, gets altered by different seizure medications, for example. You also saw a little bit of a hint of this data. So this was an example of where, and again, I heard about Fred Fay earlier. This was an example where we wanted to be able to look at the beating zebrafish heart. And, you know, everybody before then had been doing this sort of imaging where they did like a single plane and then they would do 10 beats and then another plane and 10 beats and then they get one stereotypical sort of one heartbeat beat, right? But the heart doesn't beat the same way every time. And what we really wanted to try to do here was track individual red blood cells going through the heart as a function of it beating. So we needed real-time, three-dimensional imaging. And so we had to push the system up to go to 100 volumes per second, which required using this really fancy camera that can actually image at 1,000 frames per second, which is a 1.3 gigahertz pixel rate, which is so much faster than that 80 megahertz I explained to you at the beginning. It's ridiculous. And so, oh dear, you know, I'm just going to really quickly make sure I stop my videos from misbehaving here. Okay, so yeah, so on the left here, we're looking at the red blood cells going through the heart. On the right, now we're actually looking at calcium. So this is labeling calcium in the heart cardiac wall, and we can actually watch how those calcium waves are traveling across the wall. We're imaging here nearly 13,000 frames per second, and we're only using 1 milliwatt at the sample, which is, for those of you who don't know, not a lot of light. And so working with my collaborator, Kamara Talgoff, who works in pediatric cardiology at Columbia and has different genetic models of different cardiac development, as well as collaborator in Japan, we did tracking of those cells. We were able to calculate the actual calcium levels of those cells as a function of the beat, and a whole bunch of other quantitative analysis. And that's a project now. She's got her R1 on that, and now we're inundated with fish. Worms. There's a nice worm legacy here, and I'm super excited about this one. And I'm working with Ev, who's here, and hopefully this is just going to be a very sort of brief overview. So a lot of the worm people came to me, and at the time, we'd been focusing on sort of big things, right? So the resolution of our system wasn't actually really good enough to get down to being able to do single cells in worms. But the worm is an incredible organism. It's got this, really well-detailed connectome. We know genetics. We have a name for every single one of its 302 neurons, which was shocking to me. And so we took our really, really fast camera and we set our system up, and we found that we were able to actually capture, the whole worm moving around in 3D. And so again, this was at 1.3 gigahertz pixel rate. We got this far initially, so we managed to do two-color imaging of then calcium and the red fluorophore. So then all we had to do was figure out how to track all of these cells and actually read out the neural activity from the entire brain and nervous system of a freely moving, behaving animal, which turned out to be incredibly difficult. That's nice. So not just because it's really difficult to track them, but the other thing is it's really difficult to relate what each of these cells is, which one of these neurons they actually are, right? And so that's incredibly challenging. And so I was fortunate, as I said, to meet Ev and his former mentor, Oliver Hobart, who had developed this worm that they called Neuropal. And so they made this worm, and it's the same in every single worm, and each one of those individual neurons with its own middle name has a specific expression of a certain different amount of these different fluorophores. So it has this beautiful rainbow of neurons within the whole worm. And, if we could just image that, then we would be able to know the identity of every single cell. And so Richard in my lab really set to this, and he built a multispectral version of our system with greatly enhanced resolution. So this is just a three-dimensional image of the worm that we can capture. We're actually able to capture the entire worm in all of these colors in real time, right? Which also means that this technology could be really, really helpful for all sorts of things like different particle tracking and, all these different things, but we also found that it was really, really helpful just to create the tracking algorithm, because now if we had the colors, we could be absolutely sure that we were tracking the same neuron, from moment to moment. And so we collect data like this. We actually do a color map, then we do some sequences, then we do another color map. And then Richard, as I said, spent most of the pandemic figuring out how to make this incredibly, machine learning, AI-driven tracking algorithm to extract out all of these time courses from all these neurons. And I, don't have time to talk about it all in great detail, but here's a little sort of vignette. So this is extracting neurons from the ventral nerve cord. They're going to go red. They're responsible for curvature in the ventral direction. And so you literally can see the way that the neurons in this worm are actually encoding and showing you exactly how it's controlling how its body's moving. And we've been able to now relate this to some of these classic models of how we think the worm circuit fits together. And so this is, then it was awesome, as you can then go, I wonder if there's a neuron in the brain that has anything to do with this one. And then you can just go search for it and find it, right? So we're actually, I think, got something pretty cool here. And Ev's going to be responsible for helping me make sure that gets out. Sorry, Ev. Okay. All right, adult fly brain. So this was actually, Richard Axel approached me originally to talk about whether or not we could do the fly brain. People do little cranial windows in flies. They head fix them and they put them on little balls. And so that allows them to actually walk, and you can image them, and you can give them stimuli like little odors and things. It's a really fantastic system, and compared to things like the zebrafish that usually have to be kind of embedded or and they're also a juvenile or a larval, these are adult flies. These can do a lot of very sophisticated things. They can go search for things, they can fly. I mean, you can't fly. These guys can fly, right? So we can image their whole brain in real time as they do all these cool things that we can watch. And so when Z in my lab set up this experiment, they have a really sophisticated odor detection system because, flies have to find food. They love the smell of vinegar, for example. And so he gave them a stimulus that was a sequence of different odors, right? Vinegar that they like, and then another odor that they don't like very much. And then we just, we put them on a ball, and this is very similar to the work that we do in the mice. So we image them on the ball, and then we capture all of the neural activity throughout their entire brain in real time. Now, this image doesn't look very interesting because if you're imaging all the neural activity of the brain all at once, it's like a lot to see and to process, right? So we had to come up with computational methods to actually understand this data, right? To understand what was going on. And so this is a method that we call spatiotemporal unmixing. You basically take this movie and you say, all right, I know there's a whole ton of different things going on. It's a little bit like trying to figure out all the conversations that are happening in the middle of a big crowded room, right? So first thing, if you can actually figure out sort of what those conversations actually are, then you can kind of go and see if you can then figure out where that particular conversation is, right? And so you can find these different time courses, which we do by screening the data, and then coming down to some reasonable set of time courses. So these time courses throughout this entire video represent times at which a particular pixel, basically, in this data set is turning on and off. And you can see there's a lot of cool-looking ones in here, ones that seem to have some sort of response to the odor, and some that seem to not care about the odor at all, right? So then we can do this thing that we call non-negative least squares fit. So if we know the time course, we can basically go to every pixel and say, okay, how much of each of these time courses is present in your pixel? And so we can turn that into these images of these networks. And so that fuzzy-looking, junky-looking image that I showed you in the beginning turns into this, which is a whole sequence of these networks throughout the brain that I have been isolated from that data based on the fact that they each have a different temporal property. Okay? So we're seeing that this visualization of this data throughout the brain, but it's organized in these different interconnected structures. Okay, and so what's awesome about that is now you have this sort of roadmap, right? It's a little bit like Google Maps. So you can now say, I want to look at this network, and I want to look at what this network is doing. So this is the entire odor system of the fly. This is the antenna lobe, which is basically sort of like the olfactory bulb of the fly. And then the signals that get processed initially into the antenna lobe go up to the calyx and out to the lateral horn. And so I can then extract all of those signals. I color-coded them here, and we get all of these amazing signatures of the way this neural signal is passing through the system, right? From the initial detection of an odor to then the way that that gets processed as it goes through the brain. And the thing that interests me the most is that, we all know, okay, if you're in your house and you smell toast cooking and you go, oh, toast. And then, 5 minutes later you smell toast again, you're like, yeah, toast, There's like, it's not the same. There's never anything in a living brain that is the same every time, right? But yet we do a lot of our experiments by saying, I'm just going to repeat this thing 10 times and average it together so I can see like the thing that I'm looking for. But what we're interested in is why and how does the brain change all the time? So in some cases, this animal actually ran when it smelled the odor, and in some cases it didn't. Right? And now I can actually go back and look at the data and go, what was different about the brain that led this animal on this particular trial to decide to run versus this particular trial where it didn't decide to run, right? So here I'm only showing you the odor responses, and some of them are very, very robust. So those are like the input. It's like toast, toast, toast. And then the next layer is, do I care about the toast, right? And so now I can see how that is actually changing from trial to trial because this data is, as I said, fully real time. We can look further downstream into the mushroom body. So this is more of a sort of central processing area. And we can see those same signals as they're being now transformed into things like decisions that the fly is going to run or not going to run. And then we found this awesome little structure, the protocerebral bridge, which is actually known also for navigation. It has this kind of literally a structure where you can kind of look at directions. And we found neurons in here that didn't care that there were odors at all. These neurons were just going down and up and down and up. The odors are still happening, and these neurons don't care about it at all. So well, what are these neurons doing? And I lined them all up. And I looked at what the fly was doing. And I found that those neurons were going down in the several seconds before the fly suddenly decided it was going to run, right? And so I've got these two systems in the brain at the exact same time, one of which is worrying about the odors, and another one which is worrying about whether or not it's going to run, and all of the stuff in between. So this one is not Eb's problem, this is somebody else's problem, but now we have to figure out how the fly works, because we literally have recorded every single thing that's going on, and there's even these other cool cells here that I love that just tick along and do something that we have no idea what they're doing. And, you know, we don't even see these when we average trials, right? And so and this goes back to other work we're doing in the mouse. There's so much to see here that we've been missing without having enough signal-to-noise and enough high-speed real-time information to ask questions about what on earth is this doing, right? And so we just have no knowledge of that. So then you can put the whole thing together and get this beautiful, like, multicolor picture of, you know, what's happening in the fly. Here comes an odor. You can see the way it courses around the brain. You can see the way, you can't see it. That's the point. You have to do the analysis to really understand what's going on. All right, let's take a little trip to mice. So this is a study that we published in Science in April 2020, which is literally the worst time to ever have your first paper in Science, because nobody cared, because it was in the middle of COVID. Our only saving grace was it was about odors, and everybody had anosmia at the time, and so, you know, we got some interest in that angle. So this is a collaboration. Again, talking about how collaborations happen, Wenzi was my postdoc and Lou was Stuart's postdoc, and they were married, so they snuck into the lab one night and tried this experiment to just see if it would work, and 5 years later we got it into Science, so you can't complain about that. So this was looking at the olfactory epithelium, which is inside the nose of a mouse. So these are these sort of turbinates, these kind of curved structures that kind of the air flows over when you breathe in, right? And so this structure of this tissue is it actually has neurons in, olfactory sensory neurons. We were able to express that calcium indicator in those neurons. Now, Linda Bach and Richard Axel won the Nobel Prize for showing that each of those olfactory sensory neurons expresses a G protein coupled receptor that is sensitive to a given odor. And so, and they only have one receptor. And so with all of those neurons, basically once you smell some kind of an odor, those only certain neurons send a signal up to your brain, and that's how your brain knows what that odor is, okay? So what we decided to do was look at this in this intact system and explore what happens when you smell mixtures of odors, because we knew that from a sort of psychophysical standpoint, you can smell coffee and you can smell chocolate, but if I give you chocolate and coffee at the same time, you can't necessarily tell that it's coffee and chocolate. You might call it something else, right? And so we wanted to sort of understand how, for example, one odor might suppress the representation of another odor. And so this is a sort of live video, hopefully, of the, oh no, okay, you can imagine it flashing. Sorry about that. So it's twinkling, right? And we're flowing these different odors over. And so what we can actually do with this data is there's 10,000 neurons in this field of view, and we can extract the signal from every single one of them. And we can extract each individual neuron's response to the odor that we flow over there. And then we can flow different combinations of odors, individual odors by themselves, and then different combinations of those odors. And do that for about an hour while this tissue is being perfused and is still viable. It's an acute hemi-head prep. And so then we have this massive statistics of all of these different neurons, which we did across many, many animals. And the take-home point that you know, turns out was very interesting to people was that you could have a cell that would respond to an odor, you'd add another odor in, and that cell's response to the first odor would either be higher or lower, right? Yet it didn't have any innate response to that second odor, right? So somehow or other, having that second odor there had the ability to either attenuate or accentuate the first neuron's response. And that was something that, you know, hadn't been thought of before, and something where it showed us that this kind of choice between different odors, or this mixing of these signals, was occurring actually at the initial sensory layer and not necessarily in higher processing areas of the brain. So that mouse was sort of alive, but not really alive. Going back to my origins, where I really wanted to study vasculature, this is our ability to image the volume of in vivo mouse brain at 10 volumes per second, capturing all of those vessels, and actually you can analyze throughout that 3D network the way that those different vessels are dilating in response to stimulating this animal's whiskers. And so this was, you know, this is again something that's always been incredibly challenging to do if you're just doing like one plane at a time. You can also image neurons. This is looking at apical dendrites of layer 5 neurons just kind of flashing spontaneously. These sort of like lightning movies, same thing, right? They're very difficult to interpret. We did a similar thing that I showed you on the fly brain here. We did what we call spatiotemporal unmixing. And so what that means is we're finding those individual time courses, and then what I've done here in this video is I've color-coded those neurons that have a similar time course. Now, if you separate your data here, what you can actually do is something quite fun. You can turn it into music, okay? So you can say, when this neuron fires, I'm going to play a note. Okay, so now what I did here is I organized also these neurons from low to high, right? So the first neuron that fires gets the low note, and then the next neuron that fires gets the next note, but then in the end, each neuron keeps the same note. Let's see if this works. This is a little light relief. I love it. It's just such a nice tune. This bit's my favorite. So we actually have a paper on this under review at the moment. We call it audio visualization. It's a bit of fun, but actually once you've watched your data with sound, you can't go back to watching it without. So I'm sorry about that. Big finish. Okay. We've done a lot of that. I didn't want to torture you with it, but I thought you might at this point be a bit tired. Okay, we've done 2 photon versions of this to make, to do really nice high-speed 3-dimensional imaging of the mouse brain here, capturing these 3-dimensional volumes. But, my first love, honestly, at the moment is these small brains where we can really say we're seeing, the whole brain. We've done a few extra things here. We've built a system that can image really large fields of view. So this is imaging the entire zebrafish, capturing the first circulation going through the whole fish. This is like a weird little squid that this guy Eric brought. He was great. Organoids, I wasn't sure if anybody here does organoids. This is, again, not an application I imagine that we would get people excited about, but it turns out that lots of people grow these things in these really beautiful chips where they can flow through things and stretch things and do stuff, but oftentimes they only image them dead, right? Or they sort of try to crack them out of these chips or something. Well, we found that we could just image them inside of the chip, right? And so we didn't have to worry about killing them, so we can think about now in vivo experiments that we can do in these chips, including things like looking at things like calcium signaling. And so this actually got us involved with a group that was doing radiation therapy, and so they installed one of our systems onto their beamline, and we've been using that to look at the effect of heavy ion radiotherapy on 3-dimensional tumor cultures. And so I'm learning about cell motility and mobility and calcium signaling in cells that aren't neurons, and that's actually been also super interesting. Okay, and now to living people. So this is something that we call Medscape. I'm almost done. So Creeper in my lab didn't want to work on animals. She ended up working on animals because you can't really do any of this without working on animals. But the problem that we were really interested in was there any way we could bring this technology into the clinic? Because if you go to YouTube, you can watch these videos, and I highly recommend it if you have a strong stomach. You can watch people doing robotic surgery, right? And they're like, the whole time they're going, what is this? Do you know what this is? Is this colon? I don't know. Do you think this is colon? Is this a nerve? I don't know. And they don't want to spray the area and clean it up because they don't want to lose track of how much blood they lost. They don't know anything, right? But if you want to know what that tissue is, everybody knows what you have to do is you have to cut it out, right? And you have to take it over to your lab and you have to dry it out and slice it thin and then stick it on a microscope, and then you can come back and go, it was colon. But it's too late by that point, right? We know exactly what they look like, right? They all have this beautiful structure, and so if you could actually just stick a little probe there as you were doing the surgery, you would know exactly what those things were, right? And so also this time delay, this cost, this infrastructure is crazy, right? And there's some tissues you would never dream of cutting out. You wouldn't say, I'm going to cut this nerve out to find out if it's a nerve and if it's okay for me to cut it out, right? But if you actually had a tool where you didn't have to do that, that would be great. So this was what we were sort of envisaging, and there are some technologies that allow you to get close to this, but the fact that we could do really high-speed three-dimensional imaging means that we can tolerate movement, and one of the really cool things is that our sensitivity is so high, we don't have to put dyes in there. We're actually able to image intrinsic fluorescence, so just the fluorescence of like proteins and stuff that are already present in the tissue. So this is imaging a mouse kidney, and it's inside of a living mouse. The mouse has just sort of had its kidney kind of popped out a little bit. And what we're doing is we're scrolling along. So what you're seeing, as I told you, Creeper ended up doing mice, and she wasn't happy about it. But so you can see these are just different depth planes of the kidney, looking at these glomeruli. This is the side view. And what's really awesome is if you're measuring this three-dimensional volume and you're scrolling around, you can stitch it together, right? So you can go across millimeters of tissue and create this three-dimensional block, which you can then explore basically like you would a box of slides, except that it's all actually in 3D, so you can see three-dimensional structures. And so working with Shana Coley, who's a kidney pathologist who's interested in using this to do very rapid assessment of donor kidneys. We got pieces of fresh human tissue, a human kidney, and we did, a comparison to PAS staining, showing that we could see all of these hallmarks of kidney dysfunction that she was interested in being able to evaluate very quickly. But one thing to remember here, not only are these images really cool looking, they're all three-dimensional, right? So we could always stop and actually explore the three-dimensional structure of the thing that we were comparing to a thin section. And so in this case, this atubular glomerulus, which is hard to say, we were able to really disambiguate what this was and visualize it from all angles, right? Whereas normally it's very difficult to tell what this is. And so this is another sample that we decided. This was our oh no, again, I'm really sorry. Okay, and it's gonna ruin my surprise. There we go. Okay. You can't guess what this is now because you just saw. So this is our first in vivo imaging, and it's of a tongue. Okay, so this is what happens when your IRB-approved volunteer licks the objective lens, and you capture very high-speed three-dimensional imaging, and then you stitch it all together, and you get this beautiful, gross image of all of the little fuzzy stuff on your tongue. And we did a lot of imaging. We did the lip, and we were able to do all kinds of areas of the oral mucosa very easily. And again, there's no dye added here. This is all intrinsic fluorescence. And so this is what it looks like when you cut up tongues, I don't know. You can also add a little bit of dye. This is doing a nuclear stain on a piece of just completely fresh human tissue. You can even convert that image into a basically fake H&E. So this is sort of a fresh tissue, completely not cut up, not dried comparison. And this fake H&E you can actually fly through and look at in 3 dimensions and scroll around compared to what you would get in sections. The other neat thing is if you're going to be imaging in vivo, you may as well take advantage of the fact that the thing you're imaging is alive, right? So you can actually do measurements of physiology. So in this case, we actually did a tail vein injection, and we can watch the way that dye actually comes into the kidney at this cellular level. And we did a sort of a time lapse, and we were able to actually watch the way that the different proximal and distal tubules were taking up the dye or not taking up the dye. And this could give you kind of an acute measurement of tissue function in vivo. We also did another experiment where we ligated the kidney, and we saw this beautiful wave of autofluorescence that corresponded to ischemia, that when we took out the when we reperfused, we could see it returning. And so this is also, potentially very, very useful. We can even just do things like this, imaging the microvasculature, just kind of scrolling around in 3D and capturing those images. So we're working right now on sort of establishing wider scale clinical trials, as well as miniaturizing this. So we've done some work on getting this to a smaller form factor, but we would actually really like to get this to the point where it could be a handheld device. And then really, really quickly, non-living people, I don't want to call them dead people, but in the middle of going around and showing people this system, Creeper was quite happy to be given a cleared brain, and I didn't want to image it at the beginning. I was like, that's not for us. We don't do structural imaging. Like, we just do really fast functional imaging. But these images were so beautiful that, we kind of couldn't help ourselves. And so my sort of passion project in the pandemic was that I got together with this guy, Zhu Wu, who is one of the early developers of iDisco. He's now at Cornell Weill, and he's really great at clearing human brains. And I'm like, I'm really great at making really fast microscopes. I wonder how quickly we could image a whole human brain. And so we did a back of the envelope calculation. We were like, I don't know, 10 days. And so the NIH gave us money to try to do that. So that's what we've been trying to do for the last couple of years. So we developed a system that uses this similar geometry. Juhao can clear sections that are 5 millimeters thick. So we can mount them between two pieces of glass and basically scan them really fast on my imaging system. So here's an example of us imaging one of these brain slabs. One thing that we want to do here is we have to use immunolabeling because these are humans, but we want to cram as much information in here as we possibly can. So we're going to do 9 colors simultaneously, including labeling all the nuclei, and just basically whiz around this crazy sample. It's going to be at least 3 petabytes of data per brain. So we've actually had to spend a great deal of time actually really thinking about what we're going to do with the data. What is our analysis pipeline going to be? Where are we going to put it? How are we going to give people access to it? What are people going to want to use it for when we have this data, funnily enough? But here's an example of our first brain. So this was a post-mortem brain. This was an intact 3-dimensional high-resolution MRI, which we were able to analyze to get diffusion tractography. This is a piece of that brain before it went into clearing. This is a picture of my lab through it after it's been cleared and stained by Juhao. So this is a big team effort, including people at Mount Sinai and Mass General. And then here's the image. Doesn't look very exciting, right? Because it's just, it looks like a piece of brain. But what's really nice is that you can actually take this image and you can zoom in and zoom in and zoom in and zoom in. And on any layer, you can get all the way down to the level of each individual nucleus. And this is just the nucleus channel. So this was 5 color data. So we actually had 4 different antibodies plus the nuclei labeled in here. And so we did this whole big section here, and then this section here, this was using NPY as a label, which has these beautiful filaments, and this is just to show just what level of detail we can get. And so here's another, you have no idea how difficult it is to make this pretty movie that you're watching right now, because this represents so many terabytes of data that had to be transferred to Pittsburgh and so on. But we're on track right now with this project to, we hope, image 3 whole brains by the end of the year, next year, not this year. Hopefully, yeah. So this is a whole other thing that we're doing. So, what can we learn from high-speed dynamic imaging in living systems? We can image living things while they are alive. Which is quite interesting. We can image movements, we can image blood flow, we can image cell signaling, and we can do it in a lot of different experimental conditions, like actually letting things do natural behaviors the way they want to be doing them and not trying to create these sort of complicated tasks and structures and stimulus protocols, because we can literally just capture what's happening in real time for reasonably long periods of time. We can introduce stimuli, perturbations, whatever you like, and we can also look at things like development and disease states. We can take these technologies to clinical settings and guide clinical care, and we can also use these to also do this very, very large-scale exploration of structural and molecular genetic features, again, on this very holistic scale. So with that, I just want to acknowledge my funding, my institute, and the people in my lab. This is my SCAPE team. This is my not SCAPE team. And thank you very much for this honor, and thank you for listening. There's no questions. There was too many slides.
Thank you for the meeting. I have a question. In heart cells, in the atrial sinus atrial node is very difficult to see in live preparations, and it's possible to apply this kind of technology in that preparation.
Which organism?
Rat.
So the biggest limitation of doing the in vivo imaging, and you saw this on the Metascape examples, is that you can't clear living tissue. And so if there's already some thickness of tissue, there's light scattering in there, it becomes difficult to image down terribly deep, right? So I think the deepest we've gone is sort of 300 or 400 microns. So if it's something you could access, from the surface of the heart down, to that kind of level, then, in principle, yeah, you could. Yeah, you could do it in vivo. You could do it in like a Langendorf perfused prep. I don't know, but yeah, if you can get to it, you can image it. It's like the field of dreams, sort of.